Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to the final day of MDDI's Focus on Fundamentals, Vaporized Hydrogen Peroxide Sterilization, Guidance and Considerations, sponsored by Nelson Labs and broadcast by Informa. It's day 3, and we'll be discussing toxicological considerations of VH2O2 sterilization residuals. I'm Omar Ford, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speaker, download resources, and participate in the Q&A session that takes place at the end of our presentation. The slides will advance automatically throughout the event. Lastly, if you are experiencing any technical problems, please click the Help widget found at the bottom of your screen, or type your issue with the Q&A area, and we will be glad to offer one-on-one assistance. And remember, before you take off for the day, we will conclude today's discussion with all of our presenters from this series rejoining us to address all of your questions, so be sure to stick around for that. Now, on to the day 3 presentation, Toxicological Considerations of VH2O2 Sterilization Residuals. Discussing today's topic is Lee Wentz, PhD, expert consultant at Nelson Laboratories. To learn about our speaker, please visit the speaker widget. Lee, over to you.
Thanks, Omar. Thanks for that introduction. So as you said, my name is Lee, and I'm a toxicologist at Nelson Labs. So today in this webinar, I'm going to be coming from the toxicologist's perspective with discussing vaporized hydrogen peroxide residuals in a risk assessment process, and as well some considerations for testing setups. So what we want to consider with test article selection and some conditions. So this discussion is going to be separated into 3 separate parts, starting with a bit of an overview and background. I know this is day 3 of the webinar series, but just to get everybody up to speed and make sure we're on the same page, this is hydrogen peroxide on the right-hand side of the screen. It looks pretty similar to water, but because of this oxygen-oxygen bond, it is critically different. I'm going to be denoting hydrogen peroxide with H2O2 and vaporized hydrogen peroxide with VH2O2. And it's not much faster to say VH2O2, so I'm probably just going to say the whole thing throughout this presentation. And because I'm a chemist, I'm going to be using these subscript 2s because the full-size 2s just kind of are a pet peeve of mine. So the standard that governs vaporized hydrogen peroxide sterilization is a fairly new standard. It was published in 2022, and it's the ISO 22441 standard. Now, a lot of aspects of the standard have to do with the actual sterilization parameters. So, for example, selecting a process challenge device, making sure that your sterilization process is actually effective. That makes sense. But after the sterilization is complete, some of this hydrogen peroxide that's used might condense on the surface of your device. And we also want to make sure that this chemical is not condensed in a volume or concentration that could be exposed to the patient and cause adverse effects. So this standard has a statement about the peroxide residuals, and it says, the levels of any process residues following exposure to the upper tolerances of the process parameters shall be demonstrated as being below the specified limits identified in a health-based risk assessment. And I realize you probably read that far faster than I said it, but I just wanted to say that to show that it's kind of a mouthful, and it says a lot while also not saying very much specifically. It makes sense. We want to see what happens if we expose the device to the upper tolerances of the process. So we want to see what the maximum amount of peroxide residues might be on the device, and then compare it to a value that we say is acceptable in a health-based risk assessment. So what is that value? Well, the standard simply says this health-based risk assessment should be conducted in accordance with ISO 10993 Part 17. So that means that the vaporized hydrogen peroxide standard doesn't actually give you a value. It points you to another standard. So what is the standard? Well, the ISO 10993 standards are a series of standards on the biological evaluation of medical devices. And part 17 of that series has to do with toxicological risk assessments. It turns out that this standard is actually a fairly intimidating standard. It was updated last year in 2023. It's 72 pages, and it's quite in-depth. The purpose of this standard is generally to address what they consider medical device constituents. To me, that just means chemicals. And typically, this is applied to extractable and leachable medical devices. Or medical device testing. So essentially, what typically happens is, imagine you have a polymer device and you want to ensure that there aren't impurities or residues, antioxidants, plasticizers, et cetera, that might leach from the device and be exposed to the patient in a concentration that could be harmful. So you can imagine that the standard has a whole bunch of detail and extra information than we would need if we're simply addressing one chemical, which is hydrogen peroxide. So part of this talk, I'd like to dig into what's actually crucial from this standard if we're just considering the assessment of one chemical. Now, another standard in the 10993 series that I found quite relevant is part 7, which has to do with ethylene oxide sterilization residuals. This standard has been established for quite a while and has a lot of information on the actual device selection, sampling conditions, and as well addresses the allowable limits for ethylene oxide. I realize this is a completely different sterilization modality than vaporized hydrogen peroxide, but I wanted to look at this standard as a guidance for gaining more detail and insight into what types of considerations we should have when we're performing a test on sterilization residuals. So this talk is going to be broken up into 2 more sections. In one of them, I'm going to dive into ISO 10993 Part 17 on tox risk assessments. We're going to look at how this standard selects points of departures, considers appropriate uncertainty factors, and also what is a margin of safety. We're then going to switch over to ISO 10993 Part 7, and we're going to use this as a guideline to see how does this assess an actual allowable amount. And it turns out that this standard actually contains all of the information on tox risk assessment for the sterilization residuals within Part 7. We'll then switch gears and look at the actual testing parameters and see what kind of guidelines we can gather about selecting test articles, putting them through a sterilization process, and sending them for testing. Okay, so what can we learn from ISO 10993 Part 17? In this section, I'm going to cover what is a tolerable intake, what factors do we need to calculate a tolerable intake, and what is a margin of safety. So we're not all going to be toxicologists by the end of this section, and I'm not going to be going through math and exact calculations, but I do have to put some formulas up because I want to just show what types of considerations there are that go into a tolerable intake and margin of safety, because all of that leads to this final point, which is likely what most people care about, which is why can't I just tell you a number right now that's a pass or fail value for every device, and why doesn't it work that way? So on that note, I'm going to start in the most logical place of this list, which is the middle, and talk about what is a margin of safety. So in simple terms, to paraphrase, a margin of safety would be the closest you would come to with a pass or fail criteria on a risk assessment. A margin of safety is essentially a ratio between how much of a chemical you are allowed to be exposed to versus how much is actually exposed to the patient. The tolerable intake on the top of this fraction is an amount that's calculated by a toxicologist to be without appreciable harm to health. The exposure dose in this case would be how much hydrogen peroxide residue was measured from your device. If the numerator is larger than the denominator, as in you're allowed to have more than you're exposed to, that number is going to be greater than 1. So essentially, if the margin of safety is 1 or greater, then we would consider that the device has tolerable toxicological risk. And that's the outcome of the risk assessment. So I'm going to start talking about the tolerable intake, which again is this calculated amount of how much would be allowed or considered to be without appreciable harm to health. So I just want to get everybody into the mindset of toxicology and thinking about exposure to a chemical. This right here is just a scribbled out graph on caffeine. So the toxicity of something has to do with several different factors, such as route of exposure, duration of exposure, and the different negative outcomes that you might observe. In this case, we're looking at an individual who's probably habituated to having caffeine. They might be used to having a daily exposure to caffeine. And on the far left side of the graph, if they don't have any exposure, they might actually be more irritable. As the exposure increases, they might actually become more pleasant to work with. However, at a certain point, the risk might go up. You might start experiencing some adverse effects, such as arrhythmia. Eventually, you could see more adverse effects like tremors or hallucinations. At a certain point, there might be an LD50, where there's a 50% chance of death. And then you could hit a maximum response. So typically, a dose response curve looks like this. In this case, imagine that this is an animal study where each one of these different dots is a different dose to a set of animals. So you can imagine that maybe there's 10 animals at each of these doses, and then we're going to observe over a repeated duration. So for example, it could be a daily oral dose over 28 days or over 90 days. And what we want to see is a point such as this NOEL, which is a no observed adverse effect level, which means that even at a certain dose range, we didn't observe any increase in adverse effects. At a certain point, when you increase the dose further, you might observe some adverse effects in a portion of the animals, and that would be considered the LOEL, or the lowest observed adverse effect level. Again, as the doses increase, you might observe either a higher fraction of the population with adverse effects or additional adverse effects. And this is where the graph increases to the max response. Ideally, what the toxicologist wants to find is an animal study that has relevant data, meaning a relevant route of exposure to how the device is used, and as well as a relevant duration of exposure. So for example, if you have devices like pre-filled syringes and it's going to be a single exposure, it might be overkill to be looking at 2-year animal studies for critical effects. Ideally, a toxicologist wants to find a study that has a NOEL, because this gives us comfort in that at a certain dose, there were no observed adverse effects on the population. And then from this level, a tolerable intake is calculated. So again, the tolerable intake is lower than the NOEL because we add some uncertainty factors onto this to account for an exposure to a human, as well as some different factors. And this NOEL is considered a point of departure because this is where the dose response curve begins to change shape. So that point of departure is what's on the top of this equation. And I realize at this point you might be thinking, I thought we were supposed to be talking about hydrogen peroxide residuals, and we're going into the background of toxicology. But I'm hoping that the relevancy is going to come together a little bit soon. So the point of departure, again, is something that's selected from literature, and depending on how your device is used, a toxicologist might choose a different study to reference, again, based on the route of exposure or on the duration of exposure. So that factor of how your device is used can directly affect the tolerable intake that's calculated. And then with these uncertainty factors, certain ones need to be added. For example, if the study was performed on animals, an uncertainty factor of 10 might be applied to move that value to relevancy for humans. Additional uncertainty factors might be included to account for sensitive humans. But the one that I really want to highlight is this last uncertainty factor, which can actually include multiple uncertainty factors. And some of these can vary depending on how your device is used. For example, if you have a different duration of exposure, for example, a long-term device, something that's used for a lifetime, a higher uncertainty factor might need to be included. Or if there's a different route of exposure, like your device is an implant device, but the animal study was performed with an oral exposure, another uncertainty factor might need to be applied to account for bioavailability. So depending on how your device is used for the length of time and the route, this tolerable intake could be quite a bit different. So that's one factor of this margin of safety. And keep in mind, this margin of safety is a ratio, which means that it's unitless. And that means that the units on the top and the bottom need to be the exact same. And these units are a little bit odd, but it's in how many micrograms or milligrams of a substance can be accepted per kilogram of body weight per day. And that means that your hydrogen peroxide residuals data, which is likely in micrograms or milligrams per device, needs to go through a conversion factor. And a few of these conversion factors actually have to do with how your device is used. In order to get from micrograms per device to micrograms per day, we need to account for how many devices are used. So that means that if you have a device where only one is used per day, the exposure dose would be 1/10 of something that if you had 10 devices applied. So for example, imagine you had some bone screws. I like bone screws because you can think of it as tiny devices that are likely homogeneous and multiple could be used. So imagine that versus a large orthopedic implant. Its exposure dose could be multiplied by 10 versus 1. Another factor is patient body weight. And since this is divided into the equation, that means that the smaller the patient body weight, the higher the exposure dose. So the worst case is actually the youngest patients or the lowest body weight of the patient populations that are included. So going by the ISO 10993 Part 17 standard body weights, an adult is 60 kilograms, a pediatric patient is 10 kilograms. That means that an exposure dose for a pediatric patient is 6 times higher than that of an adult. So this is where you might not want to have one set of limits for every device if your company provides a large list of device types. Something that's used in the category of one device per adult patient would then have a 60 times different exposure dose to something where there's 10 devices in the pediatric patient. The numbers that I'm talking about don't matter so much, but it's more the concept that the exposure dose could vary by an order of magnitude or more depending on how the devices are used. So that means that this margin of safety calculation, which is pretty much the output of the risk assessment, has some unique factors depending on how your device is used. The duration of exposure and the route of exposure, when appropriately communicated to the toxicologist, can affect how the tolerable intake is calculated. And then how the device is used, as in how many devices are used per day, and the intended patient populations can affect the exposure dose. So these acceptable limits can vary depending on any device that's used. My advice here is to be clear with the parameters of use of the device and clearly communicate this with the toxicologist that you're working with. It's a good idea to have an idea of how many devices is your worst case exposure per day. Because if somebody tells me we don't have a limit, well, you probably don't want to multiply your exposure by infinity. That's not going to help you out. So we need to be able to define what a worst case number of devices is to appropriately calculate an exposure dose. How long the devices must be used for is also a good thing to be very clear about. And this accounts for cumulative device use. Even if a device is used for only a portion of a day, but you might use the device weekly or daily, that cumulative exposure each day is what matters. The patient exposure route might seem obvious, but always keep in mind, if you're working for a device manufacturer, you know your device better than anyone else. So it's worth it to use a little bit of background communication and make sure that everyone's on the same page. And again, with intended patient populations, just please consider what the lowest body weight or youngest patients are, because defining this from the start really helps out with the risk assessment. Okay, so we're going to switch over to the ethylene oxide standard, which is ISO 10993 Part 7. And the first thing I want to talk about is basically looking at how does this standard discuss everything that I just talked about for the last 10 or 15 minutes. Then we'll switch gears and start talking about actual test article selection and testing parameters and see what kind of guidance we can gather from the ethylene oxide standard. Okay, so this slide is going to have a decent amount of text on it. You don't need to read everything. But the main things that I want to bring across is that ISO 10993 Part 7 just gives you a limit on ethylene oxide. It doesn't point you to another standard. It actually tells you in milligrams per day how much ethylene oxide is allowed. That being said, I want to highlight that it actually has different limits depending on the duration of contact. If a device is used for a limited duration of less than 24 hours, the limit is different than a prolonged use device or a lifetime contact device. The standard also gives you the tolerable intake in those units of milligrams per kilogram per day for each exposure duration. And it also states that the appropriate default mass for each category of special patient population shall be justified and documented. So that right there tells you that these limits change depending on the body weight of the patients. Something I want to just explain, though, is this data doesn't come easy. It actually takes 30 pages in appendices to derive these values for a full toxicological review of the literature and defining the uncertainty factors. So this isn't a straightforward thing. And one more thing is that there's also a statement about the consideration should be given for proportioning the limits downward if multiple devices with the residue of concern are used at one time. So if we have multiple devices simultaneously, the limits are going to be increased. So this tells us that all of these factors that I discussed in the first section all impact the limits that are allowed. We need to consider the patient populations, we need to consider the duration of exposure, and we need to consider how many devices are used. Okay, so now we can move on and forget about toxicology for a few minutes and start thinking about the test articles. What are we actually going to be testing, and how are we going to be testing them? So ISO 22441 simply says that the methods for determining residuals of the sterilizing agent should be selected and validated during product development. Basically, it tells you you need to do this, but it doesn't tell you how. So this is why I wanted to look at ISO 10993 Part 7 to get a little bit more insight. So the first thing is how to select a test article. We need to know what product to actually test. The standard says samples to be used for residual analysis shall be selected in such a manner as to be truly representative of the product. That sounds obvious. And in my mind, the best thing to test is always a final finished device. That's the easiest way to justify that the test article actually goes through and represents what's going to be exposed to a patient. If you have a complex device, make sure that all of the materials and all of the components are represented by the test article. If several varied materials are included, proportion of each component should be parallel to the ratio of the device being tested. Make sure that the test article is always representative. There's yet one more standard that I want to bring in briefly, and that's ISO 11139. This standard is essentially just a list of 300 and something terms used for the sterilization process. And the definition that I pulled out is the residues challenge device. A residues challenge device is an item used to assess the effectiveness of desorption. So I just liked including this term because it brings you into the mindset of not being a process challenge device for the efficacy of sterilization, but rather a device that poses the most challenge to residues analysis. It might be something that's the hardest for the residues to either decompose from or desorb from. And the reason I use the word decompose is because hydrogen peroxide would ideally decompose to water and oxygen, which we would consider quite harmless. So when you're selecting this residues challenge device, just ensure it represents all materials of the final finished device and likely is the worst case exposure. So for example, a larger surface area would have more surfaces in order to condense the hydrogen peroxide. Now, something that ISO 22441 does explain in quite a bit of detail is the idea of a product family. And this is something that can help you out if you are a manufacturer that has multiple devices of the same type in different sizes. This way, you can define a subgroup of products with similar attributes and essentially only test the worst case device. The vaporized hydrogen peroxide standard does state that product families can be used for residuals testing. You just need to ensure that the other products within the family present an equivalent or lesser challenge than the representative process challenge or residues challenge device, and make sure that you document some sort of a rationale. So this is a good way to minimize the testing burden. So now that we actually know which device we're going to select for testing, how do we need to actually handle this? So the vaporized hydrogen peroxide standard outlines that worst case conditions would be a set of conditions encompassing the upper and lower processing limits that pose the greatest chance of process or product failure. From this statement, you should consider that whatever cycle parameters you use to sterilize these test articles should be justifiable as the worst case scenario for condensing the most amount of hydrogen peroxide on the device. Then I wanted some additional guidance from the ethylene oxide standard on how to actually handle these test articles. This language is going to be quite a bit different than what's used for hydrogen peroxide, but it's more the spirit of what's said that I think is important. So some of the things that are said are samples should remain with the load of products until the day of analysis, or samples should be immediately frozen. The time between removing samples from aeration and the beginning of extraction should be held to a minimum, or extraction should be started as soon as possible after the device has been removed from packaging. If you're not familiar with how the residues testing occurs, what essentially happens is that once the devices are received by the lab, they'll put them in a beaker with a solvent in it. Essentially, just imagine you're dunking the device in a jar of water, but in a very controlled manner, at a controlled temperature for a controlled time. All of the residues that are on the surface of the device should be transferred to that solvent, and that solvent is what's then put through the analytical process. And then the concentration that's detected is back calculated to the amount of residue on our device. Now, the spirit of all of these terms to me is that the time between sterilizing the device and putting it through this testing to extract the amount of residues should be held to a minimum. If you think of it in practical terms, if your device could be sterilized and then walk down a hallway and exposed to a patient the next day, it doesn't make much sense to sterilize a device, have it sit on the shelf for 2 months, ship it in an uncontrolled way, and test it later, because that gives the sterilant a massive opportunity to decompose or evaporate, and it wouldn't be representative of a worst case patient exposure. So it's a good idea to have a plan for how to justify the time between sterilizing and testing. You can always control some of the shipping conditions. For example, you could ship the products on dry ice, and just make sure that the lab is ready to handle your products. So if they're not ready to test immediately, they could also hold your devices in a temperature-controlled manner. Okay, so now we know the device that we're testing. We selected a representative product, and we know how we're sterilizing it, and we know how we're sending it to the lab. What about testing parameters? What can we learn about this from the ethylene oxide standard? First of all, this is a very important one, which is just part of the ISO 10993 series on biocompatibility, which is if a device has no patient contact, ISO 10993 does not apply. So imagine a very complex device, now the opposite of bone screws. Imagine some sort of probe that's used in an operation that's attached to a handle that only a surgeon would use with gloved hands. And then attached to that handle is a long cable that goes to some sort of interface. It's a large multi-component device, but only a portion of it would ever feasibly contact the patient. It wouldn't really make sense to extract that entire device in a jar of solvent when only a small fraction, maybe 10%, would be exposed to the patient. So this is where communicating with the lab and making sure that everyone's on the same page with exactly how your device is used and what surfaces have relevant patient contact can help you out to not have an overestimation of patient exposure in a completely irrelevant way. We want a worst case exposure to be measured, but we still want it to be relevant. Another aspect in the ethylene oxide standard is the idea of a blank sample. And a blank sample in this case would be the same test article, but that has not gone through the sterilization process. So this is always a good idea to send in for testing, because while the tests for hydrogen peroxide residuals are quantitative, meaning that the amount we measure is very consistent, it's still not free from interference. It's non-specific in the sense that it's usually a colorimetric test with UV visible spectrometry, or spectroscopy rather. So we want to make sure that if you do end up getting a higher reading than you were expecting, it's due to the sterilization process and not other residues or chemicals which might have caused a false positive. So to summarize, make sure you can define your test article before you even start this process. And it can really help out to minimize testing needs by defining a family representative product. Make sure you use the worst case sterilization parameters before sending the device for testing, and consider what kinds of shipping timelines or conditions could be justifiable so that the residues aren't sitting and degrading for a month on the shelf before the testing actually occurs. As well, communicate with testing labs so that they know that they're expecting the product, and as well, if your device is complicated, that they'll only test the relevant patient contacting portions. And this last point is a little bit of an outlier point, but this would be a situation where, let's say you have a device that's used in neonates and you have multiple devices used at the same time. If you remember from the toxicology section, that would make your limits extremely low compared to a device that's used only in adults. And if you think you have a device type where your limits might be exceedingly small, it might be a good idea to do the due diligence, figure out what the limits might be by a toxicologist, and then ensure that the lab can meet those reporting limits. Because you wouldn't want to run into a situation where the limits of detection are actually below the tolerable intake, because at that point, the testing wouldn't really tell you anything. Or rather, the other way around, the limits of detection are higher than the tolerable intake. I misspoke. All right, so just want to bring this back to an overview and circle back to the idea of a margin of safety. So again, the margin of safety is the outcome of the risk assessment. It's the value that we really care about, and that's comparing the tolerable intake, which is calculated, to the exposure dose. And what I really want to highlight here is this statement that the margin of safety must be greater than 1, or greater than or equal to 1, and contributing factors to the margin of safety are demonstrated to be conservative, because these contributing factors are the inputs that you can help either communicating to a toxicologist or by selecting the testing parameters correctly. Now, some of these factors include the duration of exposure to the device and the route of exposure to the device, because effectively communicating with the toxicologist will make sure that the tolerable intake is derived in a conservative but appropriate manner. And then some factors that might be unique to how your device is used is how many devices are used per day, and that again can multiply the exposure dose in the same way. Make sure that you justify what the lowest patient body weight is and use that for the calculations, because that will give you the most conservative result. Now, the idea of a representative test article selection goes both ways. It's the most conservative thing to justify that you have the largest surface area and all components of construction, but also remember to be reasonable with this, where if you have a multi-component device with only a portion having patient contact, only test the parts that have that patient contact. And then finally, worst case sterilization conditions do have an impact on exposure dose. If you can justify that the worst case cycle parameters were used, that really helps with the case of showing that it's the worst case exposure. And again, justifying that timing and conditions between sterilizing and sampling the device. All right, well, thank you for listening to that overview on vaporized hydrogen peroxide residuals. Feel free to contact me if you have any additional questions that we aren't able to get through in this panel session. And I also want to point you towards the Sotera Health Academy, which has a lot of free information available on medical device testing.
Now, before we begin with today's Q&A, please direct your attention to our webinar survey, available on the right of the presentation window. If you close the survey, you can reopen the widget by clicking the icon along the bottom of your screen. Thank you in advance for filling out the feedback form. Your participation in this survey allows us to better serve you. And now on to the question and answer portion of our event. As we mentioned earlier, all of our presenters from this week have rejoined us to answer your questions. You can submit your questions for them by typing into the text box located to the right of the presentation window. If we're not able to answer all submitted questions, we'll be sure to share them with our speakers who can reply to you offline. But first, I thought we would ask Lee just a few questions. Lee, first, how are these uncertainty factors calculated?
Yeah, that's a great question. And those are outlined in that toxicology or standard ISO 10993 part 17. So for the most part, the uncertainty factors going from animals to humans and humans to humans are quite standardized, and generally those are used just as a 10. If you have specific toxicokinetics information, you can justify lower uncertainty factors. Those other factors that I was talking about that are more device specific, those are given in ranges in that standard. But for example, something used for a longer duration of time than an animal study where the point of departure was selected needs an additional uncertainty factor. So for example, if an animal study was used for 28 days or was performed for 28 days, but the device is used in a subchronic manner, there might be an uncertainty factor of 6. So generally each of these uncertainty factors is between 1 and 10. And it is outlined in ISO 10993 part 17, but it's very common to have values stack up to a total of 1,000 or so, just as a rough guideline.
And Lee, are there any working groups that are working to create standard limits like there are with EO?
I think honestly, the standard is quite new. And just seeing what needs to be added to it, I don't know how far down the horizon that is. I'm not involved on the working group on the peroxide standard, but it is a full tox review that's needed in order to actually justify these limits. And it's an immense amount of work going into part 7 for those appendices on the EO standards. So I don't have insight into how far those values are as far as a standardized and published amount. But hopefully if you work with the toxicologist, you can set up a few limits that can work with your product line so it's not a redo every single time.
And Lee, I want to ask you one more question before we bring the panelists in, but is H2O2 residue analysis currently required by FDA or notified bodies? Based on previous experience, it has not been required.
Yeah, and that's a gray area question for me because I'm imagining you were likely doing cytotoxicity testing before and just moving on with that, which is likely a far simpler pathway. All I can say is the way that the standard is written right now, it says to perform a risk assessment for part 17. So if you have pushback, the risk assessment is the way to go. If you're able to leverage historical testing or using cytotoxicity testing, that's likely a lower barrier to entry compared to doing a tox risk assessment. But again, that is how that 2022 standard is written. It guides you towards doing a residues analysis and a risk assessment.
And now, as we turn to our panel, I want to ask Helen this first question. Helen, are you available now? Can you hear us?
Sure am.
Okay. Helen, you discussed using the targeted approach to assess material performance and biocompatibility for already cleared devices to evaluate impact of switching the sterilization mode to VH2O2. What happens if there are other changes occurring as well, not just the change to the VH2O2?
Yeah, so that's a great question that I always get, like, how many changes can we make at the same time, right? And I always say when we are evaluating changes, we want to make sure that the scope of the change that we are assessing is very specified, like very specific, right? Specifically defined. Like when you're switching from something else to vaporized hydrogen peroxide, then you want to make sure that we're only focusing on that change at a time. Because we really want to use that as the variable that we're evaluating, and we're trying to tie that assessment back to the ample biocomp information that we already have on the product that is currently on the market, right? And that way we can truly focus on the change at hand, the switch to vaporized hydrogen peroxide. Now, when you have too many changes happening at the same time, you might be introducing too many unknowns or variables into the system, and then we might be effectively already causing it to become essentially a new device, and I'm making air quotes for the new device, right? So really, we want to make sure that when we're doing an assessment to assess the impact of vaporized hydrogen peroxide, just do that assessment at a time.
And this next question, I want to ask this I want to direct this towards Bryce. Bryce, you mentioned during your talk that you run testing on the package after the package after the device. Do you have a standard test procedure for various types of packages?
So there is a variety of testing that we can do for the different packages. Also, it's going to depend on when and where the terminal sterilization is taking place, whether it's single use and distributed as sterile, or whether it's going to be point of use sterilization. So the answer is we do have various tests that we can do as part of a packaging / sterile barrier validation. And we actually have a series of packaging experts that can certainly help with that and set that up. Could be a whole additional conversation, actually. But the short answer is there are various tests that are specified by additional standards for sterile barrier testing, and we actually have a whole packaging group that can help with that.
This next question is either for Lee or Jason. I don't know who wants to take this one, but what method is used to evaluate H2O2 residues?
So we use, go for it if you want to.
No, you go ahead, Lee. I think this is really more up your alley.
Okay. Yeah, so it's a colorimetric test. So essentially an indicator is used that reacts with the hydrogen peroxide. That will then turn a brighter color, and then that's what's put through a UV-vis spectrophotometer. So that's why I said it's not really a specific method, even if it is quantitative. UV-vis is very good in terms that the concentration change is linear with the light absorbance. So we're able to get very consistent results. The issue is that the indicator reacts with an oxidant, such as hydrogen peroxide. And that's why I mentioned the inclusion of a blank sample, because if there's any interfering residues, we don't want something else to maybe activate that indicator and then give you a false high reading. And then you wouldn't know if that false high reading was due to the sterilization process or something else that was already present on the device. So it's not as complex as like an extractable and leachable test where there's actually chromatography to separate separate chemicals.
And I think our next question flows right into that. It's a nice continuation of what you were just talking about. And Lee or you or Jason can take this one, but what are the acceptance criteria for residues?
So that's the whole thing. That's where we don't have that acceptance criteria for peroxide residues, and that's why we need to have all these conversations with the toxicologist to figure out what patients are this used in, so we know how to actually derive the values. How many devices are used? What patient weights are included? How long are the devices used? And then the toxicologist will search through the literature and write a document to establish the limits for that specific type of device.
Okay. And which microorganisms are used for peroxide sterilization?
Omar, can you please repeat that question?
Yes, which microorganisms are used for peroxide sterilization?
For the overkill approach, the most commonly used organism is going to be Geobacillus stereothermophilus. I have seen quite a bit of data that's also been generated with Bacillus subtilis and strains that are similar to Bacillus subtilis, but most biological indicators on the market are going to contain Geobacillus stereothermophilus, and that's the organism that's outlined in 22441.
And this next question, this is either for Jason or Bryce. Some of the VHP recipes include plasma steps. In your experience, have you seen any adverse effects on materials, polymers in particular, relating to the plasma step?
I have not. Yeah, I would just say I've not seen any adverse effects because of that additional step built into particular models of VHP sterilizers.
Sure. And our next question is, how do you determine aeration time for VHP? Is it the same way as with EO?
Well, it's going to be similar. Ultimately, we need to make sure that when we sterilize product, that we give adequate time for any residues that might be on the product to dissipate or break down. And it could be established like you would with ethylene oxide, where you figure out where that point is where your product then becomes quote unquote safe. I will say that I generally see that people are testing residues immediately following the process. So, for example, in a healthcare setting, those are going to be removed from the chamber and they might be used immediately. So generally, those samples are going to be pulled from the sterilizer and either frozen for testing or go on test immediately. So that would be a pretty common approach.
And the questions just keep pouring in. I'm going to throw this up to the panel and see who wants to take a stab at this one. But when submitting samples for residual testing, do the samples need to be on dry ice, like for EO residuals, to prevent dissipation?
The short answer is yes. That's how we typically handle it, is when we're doing the residuals. We'll coordinate with basically the chosen sterilizer and ensure that we can have the samples returned to us on dry ice in a very prompt manner, and also work with them to ensure that, like shipping day of the week, that we're going to be able to get the samples back in time for testing.
And Lee, this question is for you. Is there a typical TI or ADI that can be used for TRA?
Well, the standard, again, the peroxide standard 22441 does reference a document from the EU, which is a 200 and something page review of hydrogen peroxide. I think it's from 2003. So that does go through some point of departures, which have had some pretty extensive review. So that would be the starting point.
And here's another question that for the panel. Could you indicate the limits to apply on primary packaging materials?
We may need a little bit more information. I'm not quite sure what is meant by limits for packaging.
Okay. It's perfect, since we can always follow up and answer that later or at another time after the call. Here's another question. I believe ASTM F2475 is one of the standards for biocomp testing of packing. Could you talk a little bit about that? I believe the question is asking, is it one of the standards?
So I know that there is an ASTM standard that is working on what to consider from biocomp for packaging materials. However, from packaging materials, right, you have to kind of consider it's not really the device itself. It's mostly like what can be transferred from the packaging onto the final finished device while it sits there in the packaging, right? And those conditions from package, like in different device cases, can be different, right? Like now, if you have a dry product in the packaging, there's really less, concern of something, liquid extraction-based stuff to be deposited onto the final finished device that could then be transferred to the patient when they become in contact with the device. However, let's say if you have a primary package that includes a liquid inside and your device is that liquid, then of course we need to dive more into what can happen to that packaging or the primary packaging that then could, result some residuals or extractables from the packaging to go into the patient. So we want to make sure that we include the evaluation of packaging in the final finished device, but depending on the scenario you have, it really depends how much you need from that packaging. I hope that made sense.
No, makes perfect sense. Makes perfect sense. Lee, this next question is for you. How is the extraction medium selected, for example, for a flexible catheter?
So this is good news. I'm guessing you're asking this question because you've had some nightmares with extractables and leachables with flexible materials. And this is a different type of test where we care more about dissolving the residues and transferring the residues of just the sterilant into the solvent, so water is fine. We don't really care about the material compatibility because we're not trying to actually extract the device itself like an extractable and leachable study. So we use water in our STP, for example.
Okay. This next question, Lee, either you or Jason can take this one on, but to evaluate residues, the steps will include cytotox. And what about if that cytotox, the cytotox results are greater than 2?
If the
Lee, do you
cytotox
want to
results
take
are greater,
this, or should I go ahead?
Well, the thing is, cytotox results greater than 2 are, it's a little bit more qualitative compared to if you actually do an extraction on peroxide, you do a quantitative method, and then you get an actual number on what is the concentration of residues on the device. So if you do have a failing cytotox result, doing the actual quantitative method on peroxide residuals can give you very good insight into why the device might have failed, and then you'll know, was it due to the sterilization process? At least then you have some guidelines to either further pursue maybe your cycle parameters, or if it's not due to peroxide, to look elsewhere for that failure.
And Jason, this next question is for you. What are the approximate pressure and temperature differentials experienced during a VHPS schedule for a medical device with a large surface area? What will be the H2O2 specifications, concentrations, metals, ppms, restrictions, etc.? Wow, that was a mouthful.
Yeah. I'll address, we received a few questions related to parameters, and I'll try to address all of those with a basic answer. There's a lot of variability in what you're going to see out there. When it comes to healthcare cycles, you're generally going to see, they're similar. They're not all exactly the same. They're similar. The temperature is going to sit at roughly 50 degrees C. The process is going to occur below atmospheric pressure. It will range from atmospheric at the beginning of the cycle to a relatively deep vacuum. And that entire process from there out will occur at atmospheric pressure or lower. And as far as concentration, the solutions that we're using can range from roughly 30% peroxide. It's an aqueous peroxide solution. It can range from roughly 30% up to roughly 60%. So on the industrial side, you'll see quite a range depending on what your product is, what you're trying to accomplish. For product that say is, for example, a pre-filled syringe, you may have to develop a cycle that runs with shallower vacuums than you might with a medical device that can handle a deep vacuum. So there can be quite a bit of variability in those. I would encourage the person that submitted this question, if they have follow-up questions, to please send those my way. If we need to talk about specific cycles, I'm happy to provide a more detailed answer from there.
And we have time for one more question. And Lee, this question goes to you. Worst case could be a patient in his or her life would undergo knee and hip surgery for the exposure dose to, let us say, COCR. How would we consider the number of the devices per day?
Okay.
Interesting question.
Yeah, so to paraphrase this a little bit differently or put it in a different context, we're looking at just hydrogen peroxide residues right now, but I'm going to apply this to cobalt chrome orthopedic implants. So that context to me means that you have a device like a knee replacement, which is essentially a lifetime use device. And then on the other side, you have 2 knees and you have 2 hips. So you can imagine a patient with maybe 4 implants. One aspect here is that the peroxide residual standard references to do a risk assessment per 1093 part 17, and it is not dated, which means do it per the current version of that standard. And the current version of 1093 part 17 does not have the concomitant exposure factor that the old 2002 version of the standard did. The other aspect is that if you have a knee replaced, you're not going to get both knees replaced in one afternoon. You're not going to get both knees and both hips replaced in the same operation. So that to me is not a cumulative exposure to peroxide. Because again, the peroxide is a surface residue on these implants, and it'll likely be exposed to the patient in one bolus. It's not like, let's say that you have a pre-filled syringe where maybe a patient could get a syringe every single day. That would be an additional new exposure to peroxide daily, whereas the implant would be a single exposure and you're not realistically going to get 4 implants on the same day. So I would say just assess your device as is, and you don't need to consider these additional exposures in that situation.
And that's all the time we have for questions today. This was an amazing discussion. Thank you. We appreciate your time and expertise on today's topic. Thank you to our sponsor, Nelson Labs, as well as to everyone in the audience. We appreciate your attention and participation. Within the next few hours, you will receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may not have been available to listen to the event. Also, if you missed any of these sessions from this 3-day Focus on Fundamentals series, be sure to click the Register Now button in your viewing window. All sessions are available to access on demand. This webinar is copyright 2024 by Informa. The presentation materials are owned by or copyrighted by Informa Markets. The individual speakers are solely responsible for their content and opinions. On behalf of our guests today, Lee Wentz, and our returning presenters, Jason, Bryce, and Helen, I am Omar Ford. Thanks for your time and have a great day.